So let's say we've got somebody who has a TA4, TA3 heterozygote. Okay. And we'll imagine, so what that looks like chromosomally is you have TA4. Oops, I almost drew 4. That's what happens when I talk and I write at the same time. It's a bad trait for a teacher. And we've got a second chromosome that just has TA3.
Right.
Okay? So that's explicitly what that means.
Gotcha.
Now, in that exercise, I had said there's some random nucleotides, which is true around microsatellites. There's a unique DNA sequence that's the sequence of that chromosome, and there's just this little stutter in between. So I'll pick, I don't know, GAC and GGG to be our unique sequences this time.
Okay.
So those would be, by definition, practically present on both chromosomes. And again, come on in. That's how we know these unique sequences are how we know we're comparing the same parts of the same chromosomes. Okay. They have to be longer than 3 nucleotides for them to be unique, but that's how we know that this is that chromosome, and there's a difference in size that's just the addition of one repeat on that chromosome compared to that one. So what we were doing in class today was I had said that there's a GAC, and then they're heterozygous. The brackets don't mean anything. I'm just using them to separate the unique sequence from the microsatellites. And then there's GGG.
Okay, that follows.
So these two are equivalent molecules. So all we were asking in class is, what is the size in nucleotides of that molecule there and that molecule there? So you just count how long in numbers of nucleotides are these. So that's 3 plus 8 plus 3. 14? Yeah. And then 3 plus 6 plus 3, or 12. So those are that's the first part of that question that was in class. What are the sizes of the fragments? And then the next question is, what does this look like on an agarose gel? So if you stick both of those chromosomes into an agarose gel, there are 2 different sizes of DNA than you've put in that well in the gel. So when you apply the current, let's see, that means positives down there, and negatives up there. Then what does this look like after we apply a current and let some time pass?
There should be one band at 12.
Yeah.
And one at 14.
Right. So a band at 12 nucleotides, a band at 14 nucleotides. So it's heterozygote for a difference. They're heterozygous for a difference in their chromosome size.
Gotcha.
So we see 2 different bands because they relate to the different lengths of pieces of DNA. If this had been...
Well, we draw 2 because they're their own DNA sequences.
Right, these are 2 separate molecules. Molecule 1 and molecule 2. So they're 2 separate double helices.
Okay.
Yeah, exactly. So every person has 2 chromosomes. And if you're heterozygous like this, you've got a difference in the number of copies of a repeat. So 4 and 3, 2 different alleles, heterozygous. Then your 2 chromosomes are 2 different sizes. And that's what we can see when we electrophoresis them on a gel. And you can actually do this for whole chromosomes. It's not just chunk. I mean, you could consider that these sequences here are full chromosome sequences. Yeah. They're not. Nobody, to my knowledge, has a chromosome that's 12 or 14 nucleotides long. These are not telomere sequences, nor are they centromere sequences. So this would be a tiny little chunk of DNA is all. But they're chromosomes. And if you do really fancy agarose gel electrophoresis, you can actually separate all of the human chromosomes based on size. So you see exactly this. It's just instead of 14 and 12 chromosomes, you're talking about 200 million chromosomes and 400 million 200 million nucleotides and 400 million nucleotides. It's just a longer gel.
A lot more counting.
Yeah.
You wouldn't have to count that.
No, you don't
That's
count. You just, yeah.
Start over.
Yep. Does that help?
Yeah, no, that actually makes perfect sense.
So,
Okay.
since this is heterozygous, what would happen if it was homozygous?
Okay. So, same situation, homozygote. So let's say TA4 over TA4. So the two chromosomes don't differ in this repeat. So they're both going to be 14 nucleotides long.
Mm-hmm.
Both copies of this chromosome or part of chromosome.
So it'd be really dark at 14 twice.
Right. So at 14 nucleotides, you would have twice as many pieces of DNA. Because here, that band on the left side was one molecule of DNA. That was that one chromosome that's 14 nucleotides long.
Mm-hmm.
So there's just one molecule of DNA in the agarose gel at that point.
Okay.
So it's half as dark or half as bright, depending on how you look at the gel, literally, than over here, where you have two molecules of DNA that are the same size. So there's twice as much DNA at that position in the agarose gel as the DNA is moving from top to bottom. Yeah. And it's not always true. This is not quantitative. That is, it's difficult to actually take a real agarose gel and measure how dark or bright the bands are and estimate or actually quantitate how many molecules of DNA there are, but you can qualitatively. This one's darker, this one's brighter.
Right.
There's probably more DNA in the dark band, a thick band, than a thin band.
So does the agarose gel, does it tell you when, specific genes are present, or it's just counting nucleotides?
The only thing that the agarose gel can do is tell us size. Well, it's two things. Then it can tell you difference in size, and that's, of course, all agarose gel electrophoresis does is separate DNA by size. So it's the number one thing that geneticists do with agarose gels. And that's often, for example, in this case on the left, used to distinguish who's a heterozygote and who's a homozygote. If you have one band, like on the right,
Then it's
you're a homozygous. And this could have been for 12. So if we picked a TA12 homozygote on the right side, then they would have a single band as well. It would just be a smaller band.
Wouldn't it just have its own little square and...
Yeah, so if we... Wrong tool. If we had the TA3 homozygote and put that sample over there, there's only one band because they're homozygous. So instead that's going to be the 12 nucleotide band. This person, sorry, TA3 over TA3, yeah. So homozygous, one band, heterozygote on the left. Two different chromosome sizes, two different bands. The other thing that geneticists use PCR and gel electrophoresis for another thing is to detect presence or absence of a gene. So if you're doing that, for example, and if you don't want to hear about this because we haven't covered it in a class and it's not on the test, if you don't want to hear about it, then plug your ears or leave. I don't want to distract you from what we've talked about in class, but you could think about, we've talked about an example of a gene that's present in males and not females. So who's got SRY?
Males.
Males. Females? No. So if you did PCR on female and male samples, and you loaded those, so say the primers you use then are primers that only match and base pair to the SRY gene. So on the X chromosomes, there is no SRY. So if we depict primers as single-headed arrows, they don't have anything to hybridize or base pair to because these are complementary to parts of the SRY gene.
Mm-hmm.
And if there's no SRY gene, there's no hybridizing, there's no PCR, there's no amplification or synthesis of DNA. In the Y chromosome, though, so males have an X and a Y, which is much smaller, but the Y chromosome contains the SRY gene. So it'll have those primers able to hybridize and make lots and lots of copies of the SRY portion of the Y chromosome. So you get band over here if you're male and you're doing PCR for SRY. You get no band if you're female. So I don't know, I'm not even bothering to guess or to estimate what size PCR product that is, but let's see, female went on the left, so there'd be no band. Males, you'd have a band representing SRY. Females, no amplification, nothing.
Is there any
Talking
correlation?
PCR and gel electrophoresis.
Okay. For your class or
Yeah.
just for your lab?
Yeah, it's office hours.
Ah, okay.
First exam's on Friday.
Well, gee, that's why they're paying attention now?
Actually, they were always paying attention. They're asking good questions.
Okay.
So how would you explain hermaphrodites in that situation?
Oh goodness, so
I'm
that's
just curious.
that's developmental biology. I'm glad you're curious. That you should be as a biologist.
Is there
So-
any correlation between the males having the SRY gene and it being shorthand for sorry, guys?
No, actually,
Sorry. So women are not
I've never-
sorry.
I've
That's messed up.
yeah. Let's not go there. But no, SRY actually stands for SOX9 related on the Y chromosome. SOX9 is a different gene that's involved in sex determination.
Okay.
It's just not the master gene. It's one of the downstream genes that's involved in affecting the command that you shall be a male. SOX9 is one of the genes that's important for testis development. So this is a
Is the output of that a transcription factor?
yeah.
Oh, okay.
Right. SRY, SOX9, SOX3, all the SOX genes are transcription factors.
Okay.
So they're genes whose proteins turn on the transcription of other genes.
Okay.
So SRY is the master regulator of sex. If you have the gene and it turns on, then it goes into the nucleus and turns on the expression of or makes other genes and other proteins that are responsible for maleness. So if you don't have SRY to begin with, none of the male-specific genes ever get turned on. And this is why, unfortunately, my wife asked me about this the other day. It came up either in my daughter's elementary school class or in a Girl Scout meeting. This idea is probably Girl Scouts. This idea that female is a default sex in humans, which was once in vogue, but now is kind of outdated.
Yeah.
But the purpose or the reason that thought or that concept came up is because if you don't have SRY, then that would be the default situation. Is that if you don't have SRY, then your cells are programmed to become female unless you have the SRY gene. So only males have SRY, and well, I just don't know if it's philosophically accurate to say that there's a default sex.
Well,
Does the gene just sporadically come, or like
Well,
the
for-
second X, or
Well, that's entirely inheritance. So if you're fertilized by a sperm that has an X chromosome, then you have no Y chromosome and you're a female. You have two X chromosomes. You're fertilized if you were conceived by a sperm that had a Y
but it's
chromosome.
just random for the sperm.
Right,
Okay.
Unless a mutation, right?
right.
I have a
And so there
question
are
about that,
mutant
though.
there are mutant SRY genes that
Like trisomy?
don't. You can have mutations in SRY that make that gene unable to turn on maleness. So you could be a female that has. Let me get this right. If you are female somatically, that is, your body and your physiology are female, that doesn't mean that you have two X chromosomes. You could be XY, female, and because you have a mutation in the SRY gene that turned it off. Taken with a grain of salt, though, because if you only have one X chromosome, if you're XY, then you don't fully develop as a female because it takes two X chromosomes to do that. And that's where we start getting these conversations about intersex individuals or individuals with DSD, disorders of sexual development, who are not physically or physiologically purely male or purely female. They're somewhere in between. And often infertile.
Is that something that might be able to address earlier on with gene therapy? Because I know they've been making advancements, but it's still limited in scope as far as I've read. But I see, when I walk through the hospitals all the time, I see genetic therapist or whatever. And I mean, I can only wonder what it is they do at that level. I know for babies, they've been able to restore.
You can test, but yeah, supplying new copies of genes is still something that's in its infancy. Speaking of pediatrics.
So when it comes to agarose, if there's a ladder there, and you have the XX chromosome showing and then the XY chromosome showing, would it be different just in the position of where they show on the agarose? So is it possible to have the ladder that shows both XX and XY to refer to?
Not in this case because, and this is a problem with doing PCR analyses, there are many reasons why that sample could be blank. This doesn't necessarily mean that, and this is, again, if you don't want to know about this, close your ears. This will not be on the exam Friday. There are many reasons why PCR would not work, that is, not produce a band. One of which is, what if when you were setting up that reaction, you put the TAC, you put the DNTPs, you put the water, you put the primers, you forgot to put a DNA sample in at all.
Or you punctured it.
You just forgot. Then you get a blank well because there was no template DNA. It's like the water control.
Yeah.
It's exactly a water control, but it was unintentional. You thought you put DNA in there. You thought that was female DNA. No band. That doesn't mean that there's no S or Y there. That just means there was no DNA in the first place.
No, what I'm saying is...
So I'm just pointing out that there are many reasons to get lack of a band. So go ahead.
Like on your video, you posted the AT of the worms. You had two ladders, one that showed the ATG gene, and then the other one that showed the H something. And then when you posted the two, some of them showed the ATG and some of them showed the HTC. So is those wells just like references to see where the bands are?
Correct. Yeah.
Okay.
Yeah. So yeah, molecular weight ladders, those reference just tell you size in terms of numbers of base pairs of bands on a gel. Let's see. I've got you all here. I would like to show you. I wish I had time to do this in class, but that's an actual, what a gel actually looks like from my lab. So instead of black bands on a white background, it's white bands on a black background, but it's the same concept. With the wells you can see there at the top, holes in the gel that I pipetted samples down into the gel and then applied the electric current.
So as far as computer samples might be concerned, would it be more accurate just off the bat if they were totally mechanized without the human error involved in it? I mean, would the results be any clearer?
Not for this sort of approach.
Because we've done like limited stuff on the like beginning biology, and I mean there's a lot of human error involved in that. I know for sure. I've made quite a few.
Yeah.
But
computational is not always the way to go, though. Or at least computational analysis of visual data are not always the best way to go either. Sometimes it takes a human eye to actually review all of these data. So you can see, for example, a real DNA ladder. This one I purchased from a company. They sell it. It's called a 100 base pair ladder because they've given you fragments of every size from 100 base pairs to 1,000, and then 1,500 at the top. And I was using this to. I've got different genomic DNA samples, and this is. I wanted to show you this because this is exactly what we were just looking at. Me drawing it out. These are two chromosomes.
Or multiple of these.
That have two different sizes. It's using the same PCR primers. They're just two different populations of worms. Those are their names, HK104 and AF16, but that's not important. The difference is that one of them has a slightly longer chromosome than the other between these two primers that I'm using. And so I can tell then these were unknown samples across the top. I wanted to know, do they have the AF16 version of the chromosome or do they have the HK104 version of this chromosome? So then all you do is you just compare the physical sizes. So there, for example, I know that RH14 has a band that's the same size as HK104. So that now I know that that sample has the HK104 version of the chromosome in it. And there are a couple of AF16s, and there are some more AF16-sized chromosomes. There's some more HK104 sized, the larger, slightly larger. Right, so it's subtle. sometimes. It's not really obvious if there are... It needs to be significant sizes in chromosome, significant differences in chromosome size to be able to use agarose gel electrophoresis to determine what kinds of chromosomes. And of course, there are no heterozygotes here. These are all homozygous either for the big chromosome or the small chromosome. There's no one lane here that has two bands in it.
Two.
Yeah. So,
It's enlightening.
yeah. So
Appreciate
use this all the time
Yeah.
in my lab. Yeah. Other questions?
Well, I was going to bring out the practice exam.
Oh, yes.
Actually, yeah.
You can talk practice exam, sure.
So,
Can I just throw
for
in
a
a quick
question?
question?
Please.
Can you go and find your 2 favorite boxes of cookies and bring them for me?
Yes.
Just put me down for 2 of them.
Okay.
Oh, it is Girl Scout season now.
It is.
I just bought some
The lemonades. Oh, those are delicious.
Ah, there goes the diet.
2 boxes for... Yep.
Thank you.
Yeah, the sale started yesterday. My girls went around the neighborhood and hit all the Super Bowl parties and all the drunk guys that had the munchies, and they make a killing. I think there's a reason that they time it that way.
They do.
Okay.
Used to be one.
Anyway,
Yes, thank
let
you.
me know when you get them in.
Yep,
Excuse me.
will do. Yeah, please.
Well, thanks, Dr. Ross.
Yeah, absolutely welcome.
So I had a question about.
So this is with regard to question 6 on the practice exam. We've got 2 chromosomes. One's got 3 repeats of CAG. So we could actually just write out the 3 repeats and the 4 repeats. And then I've asked you to add 5 random nucleotides on either end. They don't even have to be the same nucleotides. They would be normally if you were looking at 2 homologous chromosomes.
That's what homologous, right?
It means the same. Homologous means it's the same. So, for example, human chromosome 1. You get a chromosome 1 from your mom at fertilization, a chromosome 1 from your dad. So your 2 chromosome 1s are homologous. They have the same genes on them normally, unless mutation. Yeah. So then all you do is count nucleotides. So you've got 5 plus 9, that's the repeat, 3 units of 3 plus 5. So that fragment's 19 nucleotides long. And this one is 5 plus 4 times 3, 4 repeats of 3, or 12 plus 5.
Making that way more complicated than it is. Okay.
Yeah, often that's the case. Not for you, but in general. Yeah. So you've got 1 chromosome that's 3 nucleotides longer because it has that extra repeat unit that the other chromosome didn't have.
Okay. For that question, they ask us for the polarity too.
Mm-hmm.
What do you mean with polarity there? Like,
You mean the 5 prime, the 3 prime?
just put 5 from 3 prime?
Yep.
Okay. And hydrogen bonds, we don't have to draw all of them, right?
I did. In fact, I should say again that I'm posting the key to this later, so I'm happy to answer these questions now, but they're also answered in the video key. I did actually draw out all the hydrogen bonds, although very poorly. It was horrible.
Okay.
Because there's a lot of nucleotides there. So
Yeah.
as long as you know that A's and T's make 2 and G's and C's make 3,
Okay.
that's the important point. So don't do it just for the sake of doing it, because you're not turning this in. So it's just for your practice.
Can I have a picture?
Yep. You're welcome. So chromosome denaturing. So who's got more hydrogen bonds, A's and T's or C's and G's?
C's and G's.
Okay, so C's and G's have 3 hydrogen bonds between them, A's and T's have 2. There's the backbone of a molecule, DNA molecule that is, top to bottom. So there's a 4 nucleotide double helix, well, 8 nucleotides, but 4 base pairs. And it's got a total of 11 hydrogen bonds. what sort of a sequence would I need to make to have 8 hydrogen bonds in 4 nucleotides?
Delete a C and a G.
Hmm?
Delete C, G. One of the C.
No, 4 nucleotides. Still
Oh.
has to be the same length. I just want fewer hydrogen bonds, so 8 total.
All AT bonds.
So for 4 nucleotides, that's the minimum number of hydrogen bonds you could have. Okay, so who takes more temperature to break those hydrogen bonds?
The first one.
So you got
More time?
more hydrogen bonds
Take more time.
means more, not time, but
More heat.
energy. Takes more energy to break all of those bonds. So think about this as like a piece of Velcro that's 11 nucleotides long, 11 inches long as opposed to 8 inches long. So the more physical contact there is between 2 strands of DNA, the more hydrogen bonds, in this case, not really physical contact, the more energy it takes to rip them apart. They're bonded, there are more bonds. So it takes more heat or more energy to break that strand than one that has fewer hydrogen bonds. And you could even add, just for completeness, you could add an extra nucleotide there, an extra base pair, 2 nucleotides, to make 10. It still takes less heat to melt that molecule because there are 10 hydrogen bonds holding together even though it's longer, the molecule on the right still has fewer hydrogen bonds than the one on the left. So it still takes less heat to melt that molecule on the right.
So the more hydrogen bonds, the more denature's gonna happen.
The more hydrogen bonds, the more temperature it takes to denature the strands. Or to melt the strands, or to break all the base pairs.
I have a quick question, kind of going back to
Sure.
the chromosomes. When
Yeah.
it says to label P and Q arms where appropriate, so for telocentric, do they even have a P arm?
No, by definition.
So-
Telocentric, there's only one arm.
So on an exam, if you were to give us a telocentric centromere and we labeled Q like in air, would we get that deduction because there's no P arm present?
No. There's only one arm, Q arm.
But that's what I'm saying. So if we put the P's in the air, like
Yeah, I
on the
wouldn't
other
do that.
side.
No, you wouldn't get deducted for it necessarily, but
Because I was trying to do.
that's the point, is to understand that the telocentric doesn't have a P arm.
Okay. But for the metacentric, is there a way to tell which one's P and Q?
Let's ask the audience.
Well, depending on, because in number 2 is, although it's metacentric, I think the one on the below, it's a little bit longer.
I don't know if
Looks
that's,
like-
I think that's submetacentric.
Okay.
It's not, the arms aren't even length. So,
So
on
it wouldn't be
chromosome number
labeled?
2, which is the P arm? The one on top or the one on bottom?
Top. You know, like a small chart.
And
The
Q
top
on
one.
top and then P on bottom.
So which one's the label for the short arm?
The P.
P. Yeah, and it's on top. So the top is the P arm and the bottom is the Q arm. There's slightly longer, the bottom arm of those chromosomes. Okay, so what about if there isn't an example of a submetacentric here, but, or sorry, of a metacentric here. Let's pull up.
How frequent?
If you look at one spot on the chromosome, what's the probability that you find a C?
1 out of 10? No.
You blindfold yourself, you grab a chromosome, and you're grabbing one nucleotide. What's the probability that you're grabbing in a C?
1/4.
Right, because it could either be an A, a C, a T, or a G.
Oh, okay. I think I'm overthinking.
So you've got a 1 in 4 chance of finding a C anywhere. What's the probability that next to it is also a C? So the probability of that second nucleotide being a C is also 1 in 4. The probability of 2 C's being next to each other is the multiplication of those two independent frequencies. So that's 1 in 16. Okay, what's the probability that the next nucleotide is a T? So by itself, it's 1/4, but that set of 3 nucleotides is 1/4 times 1/4 times a fourth, or 1/64. Okay, and that's exactly the same thing that happens over here. So you've got a 1 in 64 chance that any 3 nucleotides, if you grab 3 nucleotides that are all next to each other on a chromosome, there's a 1 in 64 chance that you grabbed an AGG. 1 in 4, 1 in 4, and 1 in 4 multiplied together. So then the trick is, what do you do with these 4 nucleotides, or 5, and whatever it is, 4 or 5, it doesn't matter. Was it 5?
Yeah.
Actually, it doesn't matter, but that's okay. I'll tell you why in just a second. So what do we do with those? The N stands for any nucleotide. So what's the probability that you grab a chromosome and you're grabbing one nucleotide and it's any of the 4 nucleotides?
It's still 1 in 4.
So 100%.
It's 1. Right, because you grabbed a nucleotide, and that's all this says, is that it's a nucleotide.
The A.
Yeah, so, sorry, a nucleotide. So any, N stands for any nucleotide, A, C, T, or G.
Okay.
So those probabilities are all 1. So you've got a 1 in 64 chance of finding a CCT, that 3 nucleotide sequence all in a row. You've got a 1 in 64 chance of finding an AGG, and you've got a 100% chance, or 1 in 1, of finding a sequence of any 5 nucleotides. Now, what I haven't, so my answer was, then it's 1/64 times 1 times 1/64. That's not entirely accurate, but it's close enough for the purposes of this exercise. And the real answer is on the key. I could calculate it right now if you want me to.
No.
But it's like 1 in 4,096 or something like that. So like 1 in every few thousand base pairs you would expect to find this restriction site.
Thank you.
Yeah.
So this question, well, I understand how you did it, but it's asking in the end for this result because you're
Yeah, what was the actual
calculating,
question? What did it say?
How frequently on average should you find a restriction site for the NU?
Right. So it's something like, I don't remember if my math is right, but it would be something like 1 in every 4,096 base pairs.
But
You would
we're
expect
looking
this
for
enzyme to
that.
cut. That's the answer.
Because you have to calculate the actual sequence itself that is definite, like this part and that part, and then implement just times 1.
Mm-hmm.
Okay.
Was way overthinking it.
Well, what about Eco R1? Now I'm going to have to Google this, because I can never remember. Eco R1 and Hindi 3 have very similar restriction sequences. It's GAATTC. Is the sequence it recognizes. So how frequently will you find that sequence in the genome?
I'm sorry, what did you say about the Eco R?
How frequently do you find its restriction site in the genome? On average, it's 1 in how many base pairs?
I think she will be 1 in a 4. No.
So
It's 4 to the 6.
right. We got a 1 in 4 chance of any one of those nucleotides being the one you're looking for. So it's whatever the answer to that is. Same answer. So the n's don't do anything, right? Because they're a probability of 1. So you can have n's, that doesn't matter. So this restriction site, an EcoR1 enzyme, on average, digests any piece of DNA into approximately 4,000 base pair chunks. Because on average, it's found every 4,000 base pairs at random on a chromosome. So with N's stuck in between or not, this is still an enzyme that recognizes a specific 6 base pair sequence. Like EcoN1, the one we started with, has 6 specific nucleotides it looks for. It just that those ones have this, that enzyme, EcoN1, requires that there be a gap of 5 nucleotides in between the sets of 3 nucleotides it recognizes. So it's really only the specific nucleotides that you have to worry about when you do these sorts of calculations.
Thank you.
Mm-hmm.